Apparatus. The apparatus used in AAS consists of a hollow cathode lamp, a flame chamber, lens, monochromator, detector, amplifier and reader.
In AAS, a hollow cathode lamp is used as a light source.
The next component is the flame atomizer, which requires an oxidant to sustain the flame, for example, acetylene, nitrous oxide or air.
The fuel along with the oxidant is added to the flame.
The nebulizer then homogenizes the sample into a fine mist that is added in the flame chamber. Let's see what happens inside the flame.
When the sample is added to the flame chamber, it includes solute and solvent particles in it. As the temperature increases, the solvent particles evaporate, leaving behind solute particles, that is, the salt. This is called desolvation. The molecules are then converted into individual atoms. This is called atomization. The rays from the hollow cathode lamp approach the atoms and cause them to be excited. Some of the wavelength is absorbed for the excitation while the remaining is transmitted and caught by the monochromator. In atomic absorption spectroscopy, only the number of rays absorbed by the atom is observed. Next, a monochromator is used to isolate the incident rays which have not been absorbed by the atoms. The greater the number of atoms, the lesser number of rays will be passed on. Once the incident rays which have not been absorbed by the atoms pass through the monochromator, it passes through a lens which focuses the transmitted ray on the detector. The detector then inputs the data into the reader, and it gives the data that is presented to it. This method is based on the Beer-Lambert's law, which states that the absorbance in a spectrometric analysis is directly proportional to the concentration of the species being observed. Let's understand what AAS is used for. It is highly useful in environmental analysis, where it helps detect the amount of heavy metals and trace elements in water and soil. It is used in the food industry for quality control and make sure that no heavy metals are present in the food or beverages. These metals may seep into food through fertilizers or other food additives. AAS is applied in biological analysis to measure metal concentrations in human tissues. The technique enhances the accuracy of forensic analyses by detecting trace metals in biological samples or residues from crime scenes.